mestrado, doutorado? Yeah, he got the master and the PhD at the Physics Applied Medicine and Biology in our department. He was very intense during his stage here. He helps a lot the physics circs of the course, and now he's research fellow and the director of the Connective Brain Laboratory in the Department of Neuroscience and Biomedical Engineering. He develops instrumentation, methods, and protocols for studying the human brain by non-invasive brain stimulation. His recent works involves the creation of multi-channel coils for transcranial magnetic stimulation, TMS, software for neuronavigation and the processing electrophysiological data in magnetic resonance image. He is one of the developers and maintainers of the medical imaging software Invesalius Navigator with the Renato Archer Information Technology Center on Campinas here in Brazil. He also participates in initiatives of scientific dissemination such as the Organization for Brain Mapping, organizing activities on neuroscience for children and youth. Please, Vitor.
Thank you, Eder. And first of all, thank you for the invitation to speak here today to all of you. It's a great pleasure for me to be back home. So I lived here for almost 10 years. As already Eder said, I did my undergrad, my master's and PhD here at the medical physics, at the Department of Physics. So after that, I went to Finland, and I've been there, living there for 5 years, almost 6 years. So it's good to get out a little bit from the cold and be embraced by the warmth of Ribeirão Preto. So I'll be talking a little bit about our development in transcranial magnetic stimulation, especially on the multi-channel device that we developed there in Finland, and all of the technologies that we have developed together to guide the stimulation, to control to process all the data that is acquired. So we still have an active collaboration with Professor Osvaldo Bafa's group here, especially with Rena, Matsuda, and Thais. So that's an active collaboration that's going on. Also, a second honor is because in 2011 I was in the 5th Symposium for Instrumentation and Medical Imaging at Porto Alegre in Brazil, so in the south of Brazil, and that was the poster I presented at the time about our neuronavigation software. So you see that a little bit has evolved from that in this past 12 years. But well, talking more seriously now, when I first met Garrido today, he looked at me and said, "Whoa, you've grown some white hair." And well, all of us are aging. And we are aging fast and developing diseases, unfortunately, and brain disease. No, it's okay. That's my pause. Thanks. So, and aging is a big problem. So that's a big problem because diseases come with that, and they can come in motor diseases or kind of more specifically brain diseases, and especially the brain disorders are very difficult to treat because we know very little about the brain, and we don't have advanced enough instrumentation for that. So that's the topic that I'll be discussing with you today. But earlier this year, so Loha and colleagues have published a paper in Nature about a paralyzed man being able to walk again, using an implanted electrode in the spinal cord and in the brain. So there's a communication between the brain and the spinal cord that was restored using these implanted devices. That's of course a medical breakthrough. It's great to see a person that was paralyzed to walk again. And how did they manage to do that? There's a certain implant, a few implants that are placed in the brain that read the brain signals and transmit that to the the spinal cord so that these signals can be transmitted to the muscles and enable the person to move. So here is an illustration of the devices, but that's wonderful, but still it's a big problem to deal with invasive technology. So there's a big need for non-invasive technology because that cannot be tested, cannot be run in healthy subjects so that the technology can improve faster. Of course, there has been some discussions that basically in the United States there has been a big problem with the companies that sell implanted devices, because think about it, you implant a device a startup is created with a new type of implanted device, and then you implant that device in the person, and then for whatever reason they stop, they go bankrupt, or they cannot manage to continue the development of the product. So basically the patients are now unsupported. So they have a device that is not supported anymore. So there's no joke on that, and that needs to be taken seriously. So non-invasive technology would be an alternative to these very difficult procedures to go. But understanding the motor system is also a very complicated thing. Basically, the motor pathways involve several regions of the brain that are interconnected, and then this signal is transmitted to the spinal cord and then goes to the muscle. All of these within milliseconds, which is kind of very fast. So we need to be able to also interact with the brain at this time scale. And if any disorder or any disease, any accident comes to the brain tissue, for example, here we have stroke illustrated, we will have a certain impairment in the motor skills, and which limits basically the daily activities of a person. This is very detrimental to the society overall. One kind of already for a long time promising technology is transcranial magnetic stimulation, in which we place a stimulation coil on top of the head. We don't need to do any surgery, and then we run a very high current through these windings, so these metal wires, and through the electromagnetic induction we can induce an electric field in the brain. And this electric field, which has, for this specific coil design, has the, can you see the pointer there? So yeah, for this electric field shape here, we can focus that in a certain region of the brain, cause the depolarization on the neurons, and that signal will travel to other regions. If we record the activity that is induced by TMS, this signal can propagate to other brain regions through the connections there, internal connections in the brain, or it can go to a certain target muscle depending on the region that it goes there. And if we kind of process this information that is recorded, we can create these maps of signal propagation and how basically these different regions are communicating. Or if we look at a certain muscle and we apply stimulation to different locations in the brain, we can map the cortical representation of a muscle at the brain. So then we can create a certain map that can be very useful to inform, for example, surgeons, or to understand brain function overall. For a surgeon, for example, we can delineate the region that is responsible for a certain function non-invasively. So this can all be done in healthy subjects or before the patient goes into surgery, which minimizes the time of surgery. And the surgeon will be able to kind of save some of these regions, and the patient can have a much better prognostic. Also, if TMS is applied in a repetitive manner, it can enhance basically the activity of different regions at a long term, and this can be used to treat some more kind of complex disorders, for example, depression, which also has been shown to be very much related with multiple regions in the brain for regulation of mood and emotion. How is TMS applied? So we have the stimulation coil, and then the operator is holding it with the hand, and then pressing a certain pedal. the pulse is delivered to the brain, and we record the activity. So basically, this system here is the state of the art in terms of commercial system. It's one of the most advanced ones. We have here the brain image on the side that guides the stimulation. And you can see that this process is extremely slow. It's like an operator needs to be there and then move by hand the stimulation coil. That can take very long time. Experiments go to 3, 4, 5, 7 hours depending on the design. As I said, it's a lot of manual work. It has a limited accuracy. It's pretty slow, so we need a few seconds to be able to manually move the location to deliver the stimulus. It depends a lot on the operator, so if you take some person on the street and hey, apply TMS on the other person, that's not going to work very nice. So there's a lot of training, and training of medical staff and of health operators is a very complicated thing. Especially in Europe, there has been a shortage of medical staff. And is blind to some of the properties of the brain, for example, the connectivity. So how can we engage with the networks and not with a certain location in the brain and be much more effective in our process? So we can basically aim at being very accurate, so on the order of millimeter. We can be fast, or we need to be fast on a millisecond scale to engage with neuronal signaling. We need to automate these procedures to make them faster and more reliable. And also we need to account for a lot of properties, well-known properties of the brain, for example, the networks. Well, the slides are a little bit distorted, but basically we have done or progressed towards that aim, developing 3 big technologies. One is multi-locus TMS, so it's a variant of TMS. The other is robotic neuronavigation that has been done in collaboration here with the group of Professor Bafa in Brazil, and incorporating anatomical and functional priors to guide the stimulation. Multi-locus TMS, so that's the latest technology that we have developed there, and it consists of combining several coils together. So now we don't have anymore just one loop of wires there, but we combine them and stack them on top of each other, and we enclose that in a box. Basically, just to give an introduction to the TMS physics, if we have the loops here, and then we put a high current there, there's an electric field that is induced in the brain. From the electromagnetic induction, we can basically estimate the electric field that is generated in the cortical tissue. There is both a primary and a secondary due to the tissue conductivity, so this needs to be taken into account. And what we do to create these coil designs is basically we are physicists, so we like to transform everything that has a complex shape into a sphere. So first of all, we change the brain, we consider the brain to be a sphere, and then we define a certain focal region where we want to deliver the electric field. And then we ask to an optimization algorithm, what is the current shape, the current distribution that minimizes the magnetic energy to allow me to deliver an electric field at a certain focal point. And with that, we can basically generate a certain, a little bit more complex coil winding than what is a conventional TMS coil. And we can kind of create different shapes depending on how we want the electric field to be designed. Now, if we apply current to all of these coils at the same time, depending on the amount of current that we apply there, it will generate a certain field somewhere in the brain. If we change the current, this field changes to a different location because it's basically a summation of these fields. So that allows us to electronically change the location of the stimulus without physically moving the coil. And we can do that at a millisecond level or sub-millisecond level. That can be done really at like 0.1 millisecond time scale. And this allows us to basically interact with multiple locations and kind of we apply a stimulus here and try to prevent that stimulus from being active on the other side and playing around with that. To generate or to design these coils, as I said, we first create a brain model. We generate a certain combination of target electric field distributions on the brain, which is our sphere. Then we create a certain geometry to compute the currents, and we optimize these current shapes to reproduce those fields that we want. And by discretizing these current shapes, we obtain the winding paths for the coil. And then we go there by hand, doing a very handcraft work, we can build these coils. But these coils alone, they don't do anything, so we need to also create power electronics to control or to deliver the current to them. In general, a TMS circuit is quite simple. It's just an RLC circuit. But for us to be able to have full control of the waveform of the pulse that we are delivering, we can use a bridge, an H-bridge with the coil in the center of the H-bridge. And we can basically use IGBTs to kind of, or closing and opening IGBTs, we can ask the current to run in different paths on this circuit. So basically we ask the current to go up and then hold there in the coil for a bit, and then it recharges the capacitor back in the circuit. And that's the current waveform that we have been using. And by generating a current like that, we obtain an electric field that looks like this. And this first phase of the electric field here, this first square, is the one that over time depolarizes the neuronal membrane and generates the action potentials. So this is how the 5-channel electronics look like. It's like a kind of complicated monster inside, but it's because there's a lot of wires connecting the components here. Everything is manufactured in our group, and we hope that in the beginning of the next year we will have one also here at the University of São Paulo. This is how it looks from the outside. It's basically the size of a refrigerator, and this is the 5-coil connected to the cabinet. The first version of the coils that we did was with 2-channel coil. Just here we have just 2 coils, one on top of each other, and that allows us to move the stimulus only along a horizontal line that is perpendicular to this, or that's basically following this green line here. And with that, we can apply the stimulus along this line and map the size of the muscle response and measured with EMG, so with electromyography. And then we can locate in the brain where is the cortical location that has highest response to or highest connection to a certain muscle. If we use 2 figure-of-8 coil shapes, we can basically rotate the field in any direction, and with that we can map the orientation sensitivity of that cortical location to the electric field. Well, electric field is a vector, so the orientation is very important. Our latest prototype has 5 channels, and that allows full control of both the location and the orientation of the field. And this is how the setup looks like in an exploded view. And that's how it looks like when it's placed on top of the subject's head. So it's a very engineering prototype version here with the tapes and all of the nice touches there. Again, only having the coil alone is not enough, so we have to have some sort of algorithm to target that electric field. So what do I mean by target is basically selecting which region in the brain we want to have the field very intense and which region we don't want to have the fields. So for that we use a very similar algorithm, which is basically minimizing the energy, okay? Minimizing the energy and minimizing the energy there, and this cost function is optimized. And with that, we can basically select the region in the brain where the field is maximum, so this yellow region, and if we want electronically change following the anatomy, the cortical anatomy. And this allows us to achieve resolution, the millimeter resolution that I mentioned earlier. And here we have like each one of these balls is a stimulus, let's say on a certain cortical location. It's not that kind of point-like stimulation, is of course distributed, but that's what we kind of illustrate here. And these yellow dots represent the maximum muscle response when I give the stimulus. So it can be very, very accurate when targeting. Whoa, the rat is escaping the circle. But jumping from humans to rats, so in TMS it's a little bit counterintuitive because it's not, it doesn't follow like it doesn't follow like pharmacological development, which usually you start from in vitro, then small animals, and then you jump to human clinical trials. Here, designing coils for small animals is extremely difficult due to several physical constraints, especially focality and also heating of the coils. But we have done quite a lot of work on also having an animal device. And the first version, and this device is to be used at the same time for stimulating and also inside an MRI. So now we can put the rat, the TMS coil, and an MRI coil and stimulate and record the MRI signal that comes out from the brain activation, let's say. But there's also another big problem. So if you look at the video here, when you put a TMS coil inside an MRI, there are huge forces involved in this process due to the Lorentz forces, the presence of the static magnetic field of the MRI, and the high currents of the TMS coil. So all of these coils that you can put there, they break. It's like a Mythbusters experiment. So a second version of the prototype, we try to reinforce, use different materials. Still, you can see a crack here in the center of the coil. But now we enclose these coil plates in another polycarbonate case to prevent any potential damage or accident with the animal. But we did a lot of these physical tests and simulations, and here what you see is basically the stress distribution on the coil plate. So here we are trying to basically understand, okay, how does these forces play a role? Where is it important for us to basically reinforce the plates and so on? So we did a few simulations, and over time, there is this shockwave propagation that reflects on the boundaries of the coil plates and then come back to the center, but basically the center is usually the victim. We applied some other basically mechanical engineering approach, adding a different layers, and we were able to basically deliver stimuli at an intensity that was enough to basically have muscle activation from the rat inside the MRI. And the MRI is now 9.4 Tesla preclinical MRI, so that increases the difficulty. The field is very high. And that's how the setup looks like. We have the TMS coil above the RF coil, the radio frequency coil, and the animal. And the bore is pretty small, so it has only 210 millimeters of diameter. So this is how the coil looks like. The MRI, the MTMS electronics is inside the shielded room. And so far we did only a technical characterization of the MTMS in this setup. So we didn't record basically fMRI from the TMS. And that's how we could also change the orientation of the stimulus and basically record changes in the muscle contraction in the animal. This was done outside MRI. Next step is actually to put everything there and see if we can record some functional MRI or some bold responses as well. Okay, going back to humans. Now humans and robots. As you can see, this stimulator, the 5-coil stimulator here, is really big. So it's basically the size of a tray, 30 centimeter by 30 centimeter. And it's pretty heavy. Like it's over 6 kilograms, and if we try to hold it by hand, it's very, very difficult to precisely put it there. But why do we need to do that by hand if there is a robot or a robotic arm that can do the job? Collaborative robots in medicine is something that's not new. So you have the Da Vinci, for example, to perform surgeries. There's also in TMS already a company commercializing collaborative robotic arms to hold TMS coils. But we don't have anything that we can actually develop on and create these new applications. These are all closed systems. And what we need to achieve is basically a setup that has a robotic arm, a sort of a navigation system where we can have the anatomy of the brain to target the desired regions, and a response monitor to collect the muscle or the brain activity with EEG and EMG. So that one operator with minimal training can actually safely run an experiment. To be able to control the robot and kind of monitor how the coil is placed on top of the brain relative to the brain anatomy, we have developed already, like for over 15 years, the Invesalius Navigator software. So it's an open source and free neuronavigation software that can be used basically to develop any application on neuronavigation. That's how the interface looks like. It's pretty much similar to many commercial systems that already exist. And it looks like a video game. So it's very fun to work with that. The only thing you need to do is to select a target in the brain. That creates you a kind of a model that you need to, that guides you, and the interface guides you on how to place the coil on the desired target. And we want also to monitor the head positioning of the patient, and sometimes placing a head tracker is really annoying, and some applications don't allow us to do that because of a certain EEG cap or some other problems. So it's interesting to have a markerless navigation possibility. So that's the benefit of having an open source and kind of easy to develop software, because you can easily add new features to that. So we use a sort of pre-trained model of facial landmarks, and basically we can, that's Rena, and basically Rena can move his head, and the webcam or the camera will track the positioning of the head, and that allows us to correct the positioning of the stimulator relative to the brain. Okay. So it also detects when the head is not visible, and so on. All of this information of navigation and targeting is also interfaced with the robot, and a certain collaborative robotic arm can then be used to kind of position the coil to the target. And by tracking the patient movement, we can also correct that location and provide a visual feedback to the operator where the coil is. And this can be done in a closed loop fashion. Combining the TMS with the robot, now putting these pieces together, the MTMS with the robot, we can basically place the robot or the coils on a certain target location, and if the subject moves a little bit, we can fix this movement with electronic targeting. So then we use the combinations of the coil to focus the stimulus on a different location to fix that movement, or if the movement is low or big, we use the robot to sort of to fix for that. So there's this kind of combined application for that. And combining both of them allows us to have very high precision in repositioning or achieving the target that we want. So here we have sub-millimeter variance, and while if we look at the manual placement, that goes on the order of a couple of millimeters. That is the experimental setup that we have there in Aalto University in Finland. And in our lab, we have basically the EEG device on the corner, and then the MTMS coils with the robot, the patient, which is always there for us to test on, and the cabinet. So this cabinet is pretty big because it has space for more channels, the navigation interface, and an emergency button in case something goes wrong. So that's very important to have when you have the robots. The robot working near a person. Doing this combined MTMS robot motor mapping, we can actually expand the region that was pretty limited before to a larger region in the brain, and we did that for a few participants as a proof of concept of the technology. But you can imagine that when I give a pulse and record the muscle response, I need to process that information somehow, and I need to process it fast to be able to actually create some algorithms that allows us to do some closed loop applications. We've developed a relatively simple deep learning algorithm that actually categorizes the MEP based on certain supervised algorithm, because we had also like a lot of MEPs that were manually annotated, and we can use this very fast algorithm, which runs in just 1 millisecond or so, to automatically process and annotate the information of these motor responses. And with that information, we can feed to another algorithm that guides the stimulator on where to stimulate next in order to automatically map the region in the brain that has a certain motor correspondence. So that is a closed loop algorithm for optimizing cortical target. That can be used both with EMG. So here we are finding the optimal stimulation target based on a translation or shifting the location of the stimulus. And then the algorithm automatically, without the operator doing anything, can find that, okay, this is the hot spot or the stimulation target. This can also be used to optimize the EEG response. We have this evoked response from the EEG, and we can basically use the same concept and optimize a certain parameter of the EEG that is measured. Now we have automated the coil placement, the stimulus application, and then also the processing of the MEPs and the mapping procedure. But this still has a limited information about the brain structural connectivity or functional connectivity and so on. So we shouldn't forget that the building blocks of the brain is the neuron, and the neurons are like various types and all connected, densely connected within each other. And then one neuron does not operate alone. It is always searching for sort of a connection, and that happens. For example, we can see here in the video, two neurons are basically kind of trying to find a connection there, and when this connection is found, I don't know what are, so if you want more information, check it out, the work by Dr. Linda Landowski. But when these neurons are connected, then the information is transferred from one to the other, and so on, and then creates this sort of network. Also, when two neurons are working together, we have something that's also called plasticity, which is basically the capability of the neuronal tissue to adapt function following a certain types of stimulus, or basically readapt or remodel and change function over time. When these neurons are connected, basically different regions in the brain are also connected, and we can use MRI techniques, for example, diffusion MRI, to map, and like after recording the MRI, we can process this image and apply some diffusion modeling to basically create a sort of map of connectivity, or of anatomical connectivity, or where these neurons or these neuronal projections are going on. And that can be done, for example, by placing a seed in a certain location in the brain, and the algorithm will kind of find the direction of diffusion of the water molecules. And that can be done, for example, in 2 stages. First, we pre-process the image, and then in real time, that information can be used to create these roads on the brain, or basically the anatomical connections. That we are starting to use. We have just finished the development of this tool, but we are now doing the neuronavigation process, or basically the coil placement, we can in real time seed or find a seed, find a location in the brain, and see where these connections that are computed with the help of diffusion MRI, where those connections lead. So now we are kind of using this additional information to actually deliver the stimulus to a region that has a meaningful connection to another region and be a little bit more kind of effective on the stimulus. So if before we just had a peeled map of a surface of the brain with the grayscale maps, now we have some much more information about the structural connectivity. Also, this has been a recent project that was supported by the Google Summer of Code, in which we incorporated the functional MRI output or overlay, the functional MRI overlay in the brain images in Invesalhos, so that the navigation will soon be also guided by functional maps that were recorded before the session. Now, this is then visualization of the whole setup working. We click on the interface, ask the robot to place the stimulator in a certain target. If the subject eventually moves the head, the robot will follow and compensate these movements and kind of cautiously adjust to these movements. And when a certain location is found, take a look here on the screen on the left side. When everything turns green, it means that we are on the target, and then the stimulation can be delivered and the treatment can start and so on. So at the current stage, we have developed a lot of the tools, and now we will start with these applications. So that goes to the next chapter, let's say, of our work. Our big aim is to have a sort of a device. So this is part of the Connect to Brain project, which is a synergy project in Europe, funded by the European Research Council, between us, a lab in Germany and a lab in Italy. And our vision is to have basically a helmet-like stimulator that covers the entire brain and can deliver the stimulus based on all of these informations that I've presented to you. However, we should be very careful, because when using all of these automated tools and deep learning methods and AI and so on, which was really nice presented in the previous talk, if we let AI think about how robots and automated algorithms actually is used for brain treatment, that's the image that it outputs. So I think this is a little bit scary, and it goes more in the matrix side of things, where the human beings are basically used just as a source of energy. This work is, of course, done by a huge number of people, which I have the pleasure of working with, especially in collaboration with the university here, and is funded by many funding agencies. So thanks a lot for the attention, and happy to discuss at any time.
Thank you, Victor. We are open for questions. Any questions?
Yeah, this is a great question, of course. Let me try to just go quickly here, if I can get there fast. Basically, the algorithm to design the coils, this algorithm that we are using, it treats all of these current shapes. Oops, I think I went too far. It uses the current shapes that are basically orthogonal vectors in a basis. So the current shapes, the coil shapes, the coil windings. So here we have a singular value decomposition in the end of the algorithm, to basically generate these components of a basis function. And because of that, these coils are, in principle, they don't have mutual coupling between them. So when we measure that, when we apply a certain pulse in one and measure what comes from the other and everything, there's very, very little mutual interaction. The problem becomes kind of annoying when we change the relative location of these coils with each other, and then they are not now anymore kind of these orthogonal vectors are on the basis. So that's kind of the trick. So to cover the entire brain, that's going to be a big problem, basically. But for these coil sets of just 2, 5, so far, they don't have this mutual inductance and so on for the TMS coils. But for the RF coil inside the MRI, there is a tuning box on the RF coil that is used to kind of adjust the resonance frequency and also kind of eliminate some of the interactions. Still, up to a few milliseconds after the pulse is given, the MRI signal is disrupted. So like, I don't know, maybe 15 milliseconds or so, we still have to wait, and then after that, the signal, RF signal is basically back to normal. But that's kind of the characterization that we did so far.
More question?
Lucky guy, because it's a very different. The problem that you present here is the difference between physics and engineering. In physics, ideally, you reduce is a minimal inductance, mutual inductance between them. But in practice, when you apply a very high current that your case, it's almost impossible to reduce the interaction. But if you say that.
Yeah, we measure them like connected, disconnected, and so on. And then in the algorithm, we create a matrix of mutual inductance to compensate for, and that is taken into account in the targeting part. There is a little bit, but not that big.
Another curiosity about the geometry of the coils. Why are you still using a planar geometry for the coils?
This is just for ease of manufacturing at this point, but the latest prototype that we are building now is curved. So we are now going for a curved geometry that is basically following this geometry here. That was done because it was the simplest one for us to build very fast. The curved is way more promising in terms of coupling to the brain and so on. But yeah, that's the next one.
Another question? Please.
Oi, Vitor. Aqui da cabine técnica. Primeiro, parabéns pela apresentação. Nós temos uma pergunta aqui da transmissão online.
Estou procurando aqui, cadê?
A pergunta é sobre um dos gráficos, mais para o começo. Quando você mostrou que a corrente é aplicada nessas bobinas, o eixo estava em kA e no gráfico os valores estavam entre 2.000 e 4.000. Aí ela pergunta, o fio é supercondutor para suportar valores tão altos?
Não. Não precisa ser supercondutor, porque o pulso é extremamente curto. Ele dura cerca de... okay. So basically the question is if there's a need for superconductor wires to withstand the high currents. So yes, the currents are on the order of kiloamperes, but if you look at the time scale is very, very short. So it's just a few tens of microseconds that the current is actually running through the coil. So there is some power dissipation, so there is some heat generated and a little bit of loss, which can be seen from the kind of decay that we have here in the top of the coil current waveform, but that's not a huge issue, kind of, that would make us require superconductor wire. Here what we use is a Litz wire with kind of 40 strands. So this wire here is basically a Litz wire with many fine strands, but that's it.
Victor, yeah, I have a question here. First of all, thank you for the very nice talk. I have a question regarding the red coil. you said that you have problems with the strength, but what about the heating of the coil? Do you also have problems with that? How many pulses can you give in a row?
Well, they all broke before we gave too many pulses, so. but it's a bigger problem than the human because they hit a lot faster. We didn't measure actually how many pulses we actually give before that. So that's still on the to-do list to be tested.
One more question? No? Oh, Victor, I'd like to ask about the inductance, but about how the electronics works, and then with this very short pulse, but you have a significant inductance. This will drive some challenge, about the electronics to control these pulses for you move on along your vents.
Yeah, so the coils, they have inductances on the order of a couple of tens of microhenries. So they are usually like 15, 18, 20 microhenries. And of course, when you are switching these IGBTs very fast, there are spikes that appear in the circuit. And if you see here, there are some white small boxes on here right below these green PCBs. So these white boxes are snubber circuits that are there to dampen these spikes generated by the fast switching of the IGBT. So there are some kind of electronics adjustment to kind of prevent these high voltage spikes that would eventually break electronic components and so on. But yeah, that's usually the order of the coils have about 20 microhenries and like 100 or 90 milliohm of resistance. That from the coils, that's what I know.
Okay, I would like to thank Victor, who came from Finland to Ribeirão Preto.
Thank you very much again.
Só gostaria de aproveitar aqui que a gente teve uma notícia, já que está o Renan e o Vitor. A tese do Renan, com orientação do Vitor, orientação do professor Bafa, ganhou o prêmio de melhor tese da USP na área de inovação. Então, é exatamente sobre o que o Vitor estava apresentando, uma parte dela. Então, parabéns aí ao Vitor e ao Renan, estão aproveitando para fazer. Legal demais? A gente acabou de descobrir aqui. Então, agora está no nosso horário de almoço. Quem quiser fazer aquele tour guiado que eu falei para vocês pelo campus, tem lugar ainda, né, no nosso micro-ônibus, e às 2 horas, né, às 14, a gente volta para cá para a próxima palestra, tá ok? Obrigada.